Well, welcome all of those who have joined so far. Welcome to this really fascinating webinar on rescuing the immune system for cancer treatment and prevention. We have a big treat here today to be hearing from two of our Find the Cause Consortium, Research Consortium scientists, Dr. David Scher and Dr. Charlotte Cooper-Wasser. I'll just give you a brief rundown on each scientist, and then we'll turn it over to Dr. Cooper-Wasser to start with. So we're going to start with Dr. David Scher's little CV bio that Dave, I put something together. I hope you're okay with it. So, Dr. Scher is the chair of our Find the Cause Research Consortium. So he's a molecular biologist, toxicologist, and cancer immunologist, and an internationally recognized expert on the aryl hydrocarbon receptor. He received his doctorate from Cornell, did his postdoctoral work at Harvard, joined the faculty at Harvard Medical School, and then was recruited to BU as a full professor, where he recently became an emeritus professor. So congratulations, Dave, on an amazing, amazing career. We're really happy for you. So Dr. Charlotte Cooper-Wasser is a longtime member of the consortium. She's the director of the Convergence Laboratory of Biomedical, Physical, and Engineering Sciences at Tufts University School of Medicine, where she's also a professor of developmental, molecular, and chemical biology. She's an internationally recognized expert in the fields of mammary gland biology and breast cancer prevention. Just have to mention the rest of this because it's so impressive. Following her doctoral studies at University of Massachusetts Amherst and prior to her current work at Tufts, Dr. Cooper-Wasser was a Jane Coffin Childs postdoctoral fellow at the Whitehead Institute for Biomedical Research at MIT, and has been recognized for several prestigious fellowships and awards, including the Howard Hughes Fellowship, the Merck Fellowship, and the COG/Adventist Young Investors Award, and the Natalie V. Zucker Award. So they're both so completely impressive, and we're so fortunate to have them here with us today. So I'm going to turn it over to Charlotte, and we will get started.
Well, thank you, Drew, for that lovely introduction. And also thank everyone for being here at this webinar. Especially, I want to kind of note and call out the Prevention Party Committee that is in attendance here. Want to thank you for being here, but also really want to thank you for your hard work, your commitment, and your support for making the science possible and for making this webinar possible. So I'm going to share my slides. Is that working? Yes. Wonderful, great. So David and I are here together to talk to you about how the immune system protects us from cancer, so it can be harnessed and to prevent cancer in the first place. And so, by way of background, the first question that comes about is, how do we even know that the immune system plays a critical role in preventing cancer? Well, evidence comes from patients with weakened immune systems. For example, those with HIV/AIDS have about a 20 to 100-fold higher risk of developing cancer compared to the general population. Also, patients and individuals who take immunosuppressive drugs because of following organ transplantations are also about 2 to 4 times higher developing cancers than the general population. These individuals are also especially high developing cancers related to viruses. And so these tumors tend to be of non-Hodgkin's lymphomas, capsular sarcomas, and even invasive cervical cancers. However, they're also at risk of non-viral cancers, especially skin cancers, a specific type of skin cancer referred to as squamous cell carcinoma. They can be up to almost 250 times more likely to develop those type of cancers than the general population. So this actually kind of tells us that our immune system is essential at suppressing and even preventing cancer. But the immune system needs to be finely tuned and balanced. Too little activity and cancer cells can actually slip through unnoticed. Too much activity, particularly in the form of chronic inflammation, can also lead to cancer. hyperactivation of the immune system also has the potential to lead to autoimmunity. And so therefore, it's a key. The immune system is a key balanced to be able to recognize threats and eliminate them, but also you don't want it to be overreactive, so that way it can actually lead to pathologies like cancer or autoimmunity. So how does the immune system work in healthy people to prevent cancer? Well, immune cells are constantly moving through the circulation where they interact with everything in the body. Their job is actually to identify and eliminate those threats, whether it's an infection, aging cells, or even early cancer cells that otherwise would escape the immune system. Most people realize, or actually don't realize, that the immune system has its own connection network, the lymphatic system. It's a system that's constantly networking information across the body through the lymphatics and the lymph nodes, through surveilling of danger signals. So you can kind of think of it as the body's internet system. The lymphatics are actually the cable, the data cables driving the information everywhere, where the cells circulate, look for threats. And the lymph nodes are the parts of the body where the kind of act like routers or firewalls, where they kind of check everything that's coming through. So all the sort of filtration of the immune cells goes through these lymph nodes, and it filters out anything suspicious and decides whether or not there's any threat that's around in the body. And if there's anything suspicious, it alerts the immune system to actually kind of mount an attack. And so this will mobilize other cell types and other defenses. And it's similar, as you can think of, as kind of like the immune system and how it kind of surveils everything and remembers what's been tracked. And so it can respond faster next time. And so as you can see here, there's actually many different types of immune cells that are involved in surveilling danger signals. You know, you've heard of them. They're dendritic cells, NK cells, all these sort of players. They're involved in communicating with different cell types in the body and different cells and tissues. And they work together in a very controlled manner to communicate what's going on in the environment with the other immune cells in the body. You can kind of think of it as like a sports team where you've got different players.
You just got muted, Charlotte. Yo.
Oh, I'm muted. How much of that did you not hear?
Well, we
All
did
of
last
it?
half hour. Just kidding.
Just about 2 sentences.
So I was basically just kind of referring to the fact that you've got a team here of players, and they all have different roles on the team. And their job is to kind of communicate and sense what's going on in the environment. But there's really three main kind of MVP players on this team, and those are the macrophages, and then the lymphocytes, the B lymphocytes, the B cells, and the T cells, right? And so what do these guys do? How do they mount a response? Well, the macrophages tend to kind of be the first players on the team. They're kind of the surveillors. When a pathogen enters the system, whether it's a virus or a microbe, or I'll talk about later an antigen that's presented, these cells actually will gobble up the foreign material, will digest it, break it up into pieces, and then will actually reflect it onto the surface of the cell or present the antigen onto the cell. And this is a way to kind of allow the T cells and the other players on the team to recognize if anything is foreign or unusual. And so when a T cell recognizes the presentation of this foreign material from the macrophages, it actually becomes activated and then has kind of two functions. One function is to become a killer cell. And so an activated T cell can decide to become a killer cell, and that killer cell then has the capacity to go and actually kill an infected cell. The other type of lymphocyte that gets activated when it recognizes a foreign object or a foreign pathogen is to become a helper cell. And these lymphocytes, their job is to help the other immune cells become activated, and in particular, the B cell. And so you've got the killer T cell, and then you've got the B cell as kind of your true kind of fighters in the immune response. The B cell will actually expand, grow out, and actually become what's referred to as a plasma cell. This is a cell that really just is basically an antibody factory. It just pumps out and produces antibodies that are specific to that foreign antigen, that pathogen, that microbe. And so those antibodies then go throughout the circulation that will bind up all the invaders and all the foreign material. And then your macrophages come back again and gobble up whatever's been killed off or what's been bound up by the antibody or the dead, dying cell that the T cell has killed. And so this is really kind of the core players of how an immune response and reaction takes place after a microbe invasion, a bacterial infection, a viral infection, or something that's considered foreign. And so this process actually is a very well-controlled process. There's that kind of primary response where it recognizes something as foreign, and then it mounts the immune response. You get all this sort of expansion of your T cells, your B cells. But then when everything's kind of cleared away and everything's kind of over and your immune system has won, actually it's not over. There's actually a reservoir of cells that stay behind after the fight. And those cells are referred to as memory cells, and generally memory B and T cells. And these actually stay around for decades, sometimes even longer. And it's actually what is referred to as memory or long-term memory. And this is why natural immunity is so potent and so protective, because you now have players after this game that are now sitting around in your system ready to be called upon the next time that infection comes through your body. And because they're already primed, because they've already seen this material, this foreign entity, they're able to respond very quickly and mount an immune reaction very quickly. So then what may have taken 1 week to 3 weeks to respond to now would only take a few days. And so having this memory is actually very important for long-term immunity. And this memory can come from both a natural infection, and those memory cells tend to be the most long-lived, but they can also come after a vaccine reaction as well. And so building that memory is actually key to building a robust long-term functioning immune system, and allows you to actually maintain that protection for a long period of time. How do the environment, what are the effects of the environment on this process? Well, we know that pollutants, we know that endocrine disrupting chemicals, we also know that very chronic infections can all lead to perturbations and disruptions of the immune system and the process we just talked about. And it can lead to disruption in a number of ways. One way is that it can actually create an overactive immune system, like I mentioned to you ahead of time or earlier, that can lead to autoimmunity. It can also lead to an overactive immune system in the form of chronic inflammation. And we know that chronic inflammation is actually an important driver of developing cancer. And we also know that these chronic exposures to endocrine disrupting chemicals, as well as other pollutants, can overall weaken and kind of immunosuppress the system as well. It depends on the chemical, it depends on the exposure, how long, but this is a very important area to study that's understudied in general, and especially in the context of cancer, because what I'll share with you in a moment, being able to finely tune the immune system to be able to detect and kill off cancer is quite important for prevention. And so if these chemicals are directly influencing that ability, then you clearly have a target here that we need to eliminate or we need to understand in order to counterbalance the effect these chemicals have on the immune system. So one of the major considerations that the immune system is constantly dealing with is being able to understand and identify the difference between foreign and self. And this difference is important because if the immune system doesn't recognize self properly, this is where autoimmunity comes from. The body starts attacking itself. And if the body doesn't recognize foreign properly, this is where infections and pathogens can also become deadly because these things will overrun the system. So how is the body able to kind of identify self versus foreign? And so this is really based on, I mentioned earlier, this property of cells being able to present antigens or present proteins on their surface for the immune system to then surveil. And there's a marker on all cells called MHC. This is kind of a badge for the cell, so to speak, to say, I am self, a self badge, a self-identification badge. And so when the immune cells see this on the surface and recognize that badge, they say, okay, we recognize you, we're self, we're not going to attack you. If, however, they either present something that's abnormal on that receptor, or like pathogens, they don't even have the MHC molecule, the immune system will attack it. And that's kind of the flag that it uses to be able to distinguish between foreign and self. And so in the context of cancer, this is very important, right? Because in cancer, this is a process where we're taking our normal cells, self, and things are going wrong. And so then how is the immune system able to identify that difference, right? The self is self, but self has gone a little bit off the rails, and you want to be able to kind of check it in and keep it from forming a cancer. Well, this process is called immunoediting. And it's a process where, like I said, normal cells will become changed over time, whether it's through exposure to carcinogens, radiation, chronic inflammation, right? These things all kind of promote DNA damage, instability, changes to the genome. And so you have cells here that are essentially kind of precancer or cancer cells. And I think we've talked about in other webinars and other contexts that we are all walking around with cancer cells in our body, especially as we get older. This is just kind of a normal process of aging. You acquire mutations, but not everyone's walking around with cancer. And the reason is because of this process that is really important in kind of controlling the abnormal growth of self. And so the way this happens is during the process of early cancer or cancer, you'll have cells that have alterations in their DNA mutations. But as I said, because these are mutant proteins, these are not naturally occurring proteins. They are created because of the DNA mutations that's been sustained over time. They get presented on the surface. These are mutant proteins. These are not self proteins because they're altered in some way. And so the immune system recognizes that as foreign, right? It says, oh, this is not normal me. Something is wrong here. And so it actually mounts localized immune reactions using the T cells and the macrophages that we talked about. And so they eliminate all these kind of early cancers or even really small cancers because of that ability to kind of display and present these mutant abnormal proteins. And so this happens all the time. It happens every day in everybody, and this is a way to kind of keep cancers at bay. Over time, however, cancers will keep growing. There will keep being this turnover, this trying to actually grow out. And so eventually a cancer will come out of this elimination phase just by stochastic numbers. You have enough cells to turn over, one will escape, and will then kind of grow into a very small tumor. And this next stage of immunosurveillance of these cancers is called equilibrium. And so this is where you've got a couple more mutations, a couple more alterations. And so these cells are getting a little bit more sophisticated in their ability to evade the immune system. But the immune system still has some ability because of the factors that are being secreted to still recognize it and keep it from forming a full-blown cancer. However, as cancers with time, and especially with changes to the immune system, these cancer cells eventually escape. And the escape process is a little bit more sophisticated than just, the immune system has stopped working, right? So the immune system has stopped working, but why? And it's because a couple of different things. One, the cancer cells themselves now express proteins that actively suppress the immune response. And so they literally are sending out signals to tell the immune cells that are around to stop activating properly. And some of these, and then other proteins are expressed on the immune cells themselves in a way that allows them to no longer interact properly and mount an immune system. And so some of these actors, I'm going to present them here because Dave is going to talk to you about their importance, is the PDL1 protein, which is a ligand, which is basically a protein that's kind of expressed on tumor cells, and its receptor PD1 in the immune cells, and then CTLA4 as well, a receptor on the immune cells. And these three players are the essential cornerstones for evading immune suppression in cancers that can actually then be exploited and used therapeutically to actually help fight against cancer. And so with that, I'm actually going to punt this over to Dave, where he's going to talk to you about how the immune system now can then be used to treat and prevent cancer, which I hopefully you can take from this as a real optimistic direction in the future of cancer research, but also therapy.
Okay, y'all can see my slides, right? Yeah,
Yes.
good.
Yes.
Uh,
We see, there you go.
Yeah, okay. Yeah, thanks, Charlotte. So, I mean, now that Charlotte has given you Immunology 101, I can talk about how the immune system then can be exploited to try to fight these cancers and prevent them. So this is a trying to get rid of one thing, one moment. Okay. So this is an outline of what I'm going to talk about over the next 20 minutes or so. I'm going to talk about three general topics: monoclonal antibodies and what they are, designer T cells, also known as chimeric antigen receptor or CAR T cells, and some cancer prevention. We'll talk about vaccines and ways to maybe avoid cancer. Now, monoclonal antibodies, I'll talk first about their direct effects on cancer cells. There's a lot of monoclonal antibodies now used for cancer and other diseases as disparate as eczema, autoimmune disease, arthritis, lots of other types of illnesses and syndromes. You'll see these things advertised in TV every night, even when you're on Netflix, because it's a commercial and that's annoying. If you see them and they have an ending that says MAB, that means they're a monoclonal antibody. So there's this whole class now that have been developed in the last 15 years to treat lots of different kinds of diseases. So I'm going to talk about the direct effect on cancer cells and how these monoclonal antibodies were first discovered or generated. So here's three guys that won the Nobel Prize in 1984. This is Cesar Milstein, who was an immigrant from Argentina. George Kohler, shown here in his obviously college yearbook photo, and Niels Jerne. They started in '74 with NIH-funded money. They won the Nobel Prize 10 years later, but the first application for these monoclonal antibodies in cancer didn't happen, or in anything, didn't happen until 1997. So I just want to put a plug in here for the NIH, because this is 23 years where there would have been no support other than the NIH, which is dedicated to building things that have some potential. So how do you make these monoclonal antibodies? So Charlotte just told you that if you take some foreign protein and inject it into an animal, so this animal's never seen this protein, let's call it some human protein, it's different than the mouse. The mouse will make these antibody-producing B cells. And when Charlotte says they make antibody, they make 2,000 molecules per second of antibody. And now you'd be making antibody against protein A, but that's still not enough to treat a human. So what basically Milstein did was he took those cells and literally fused them together with immortalized cells that were a form of cancer. This is all done in culture, so not a cancer problem. So now these hybrid cells have two properties. They're producing bucket loads of antibody, and they are immortalized. So you can grow them in culture, basically in vats, forever. So that's why they're called monoclonal, because it's one cell that is turned into a clone of lots of cells, all of them identical, all of them making the same antibody. And now you have this monoclonal antibody against protein A. Now, you may not have heard Herceptin, also known as trastuzumab, which was the second monoclonal antibody approved for cancer treatment, I think for any treatment. And Herceptin is an antibody that is specific for HER2, and you know that HER2 positive breast cancers have got this protein on this surface. It's actually a growth factor receptor, so when it's activated, it tells the cancer cell to divide. So what they did, what several people tried to do and eventually generated Herceptin, was to take the HER2 protein, which is on the breast cancer cell. So here's my representation of HER2 cells. I know, very sophisticated drawing. And this mouse will make antibody against HER2. You make bucket loads of it. You have that. You infuse it back into the patients. And now it turns out there are at least 3 ways in which this Herceptin can eliminate the HER2 positive breast cancer cells. The first is it just covers up the receptor. And I just said that this receptor is a receptor for a growth factor, a hormone essentially, that tells the breast cancer cell to grow. So when it's blocked by the antibody, the activator of the receptor doesn't get in there. The second way it kills is when the antibody binds to the receptor on the surface, the HER2 growth hormone receptor protein. It initiates a process whereby the whole complex is internalized into the cell and then gets degraded. So in the second case, there's less HER2 on the surface. And the third way is that the antibody can form a bridge between the HER2 protein on the cancer cell and other immune cells, which will now gobble up these cancers. So very efficient killing and elimination of the breast cancer cells. So how good is it? This is a very early study from a very early study. And what they were looking at here is survival of patients with HER2 positive breast cancers. Now, in those years, most of these studies were done with people with advanced stages of these cancers. So this is sort of an understatement of what Herceptin can do. If you give just chemotherapy, at the end of 4 years, 67% of the patients are alive. If you give chemotherapy with Herceptin, 85% survive. I'd rather be in that group than the group down here. So that's pretty good, but you can make the Herceptin even better. And now, just to bracket this whole thing, I'll show you the results of a study that was done last year, or published last year, where what they did was to take Herceptin and put basically a poison or a chemotherapeutic onto it, conjugated it, basically making like a silver bullet. So in this case, they've taken the HER2 protein, they've made the monoclonal antibodies, but they're adding this chemotherapeutic known as emtansine onto the antibody, silver bullet, then you give it back to the patient. The antibody targets the chemotherapy directly to the tumor cell, so you have less off-target effects than the rest of your body, and then very efficiently kills these tumor cells. How much better is the chemo-conjugated Herceptin than Herceptin alone? Here's this very recent study. This is the curve of survival that I just showed you. 84 months, 67% are alive with chemotherapy plus Herceptin. But if you add the chemo conjugate to the Herceptin, so it's now a silver bullet specifically dragging the chemotherapy to the cell you want to see affected, you have 80%. I'd rather be in this group. So very powerful function. Those are some of the direct effects on cancer cells. Now, Charlotte briefly touched on these suppressors of the immune system called immune checkpoint inhibitors. That's what we're going to talk about next. Those were pioneered in part by these 2 guys who won the Nobel Prize in 2018. Jim Allison, who discovered this protein that Charlotte mentioned on T cells called CTLA4, stands for cytotoxic T lymphocyte antigen 4, if you care. And Hanjo discovered this pathway or illuminated this pathway between programmed death protein 1 and programmed death ligand 1, where this is the ligand or the activator of this receptor. Again, their work started in 1992, NIH-funded work. Approval of the first monoclonal antibody targeting these 2 proteins came in 2011, so that's what, 19 years of NIH support to get there. They won the Nobel Prize in 2018. So how does all this work? This is a curve of an immune response similar to what Charlotte had shown. So if you're exposed to some virus or some foreign protein, whatever it is, you get this immune response building over time. Now, during that period, your immune system is hyped up. There are and Charlotte mentioned this, these macrophages, which literally in Latin translates into big eaters. And they eat things like viruses and bacteria, take the proteins, and then put their foreign proteins out on the surface to present them to T cells. Now you have T cells with millions of specificities, meaning they could see millions of different foreign things, but there's only a few, literally maybe a dozen or 2 dozen cells that have got a receptor that can see this particular foreign protein. If this T cell finds this foreign protein on a macrophage, it tells the T cell, I think I'm seeing something here. There's a foreign protein, but it doesn't get started. It doesn't actually start doing its killing or dividing or making various hormones until it gets a secondary signal from the macrophage, which is this protein called CD80 that activates the receptor on T cells. So of course, the names of these things don't matter. All you need to know is that this is a go signal, and only T cells that have seen their specific targets will be the ones to receive that cell and to go. So that's the go signal. Now, during the second phase of this response, hugely inefficient, use of too much energy, the immune system to keep going when the antigen's already been cleared, the pathogen's already been cleared. So there's this suppressive component that actively shuts it down. And that's mediated by a couple of these proteins, which Charlotte mentioned, known as immune checkpoint. So immune checkpoint means inhibitor of immune responses. The macrophage will then put out this molecule called PD-L1, and the T cell will put out this receptor called PD-1. And the interaction between these two says, stop, do not respond. The T cell also will make the second inhibitor, so second immune checkpoint, because there's always some redundancy in biology. And that now sees this guy and then will suppress the cell. All this stuff not really important. What's important is once you start expressing these immune checkpoints, the T cell no longer can expand. Now, cancer cells, being the bastards that they are, have sort of figured this out. They figured out that if they put out PD-L1 on the surface, they can tell the T cells to shut up or to die. So that's what happens in many of these breast cancer cells, even though you have HER2 here and some mutant proteins, the PD-L1 tells any T cell that might be able to see anything on the breast cancer cell to just shut up and not respond. In addition, breast cancer cells produce factors that upregulate this CTLA4 on T cells, also a second immune checkpoint. So these two things together really tell the T cell to shut up. And you can tell when this is going on in cancers because you can look at the cancers, and the ones in which this is happening, you find very, very few T cells because they've been killed or they have been inactivated and they're just not homing to that region. The kinds of tumors at which you get a good immune response, you can see these T cells infiltrating, and they figured out ways to bypass this. Now, the place where these immune checkpoint inhibitors come in is monoclonal antibodies that can see either this or this as part of this negative signal, or see CTLA4. So antibodies against PD-1, for example, called pembrolizumab, will block the PD-1 and allow the T cell to attack the killer T cell, or the breast cancer cell. On the other end, there's a second antibody called avulimab, which is an anti-PD-L1 monoclonal antibody that will just cover the PD-L1. So these are almost redundant, but not quite for nuanced biologic reasons. You also have this antibody, one of them called ipilimumab or Yervoy, against CTLA4, and that will block the negative signal that occurs here. Now, this is amazingly useful for the following reason. Here's a advertisement for Keytruda. This is what I see all the time on Netflix, and it's pretty hilarious in a sense. Here's I guess the wife, and she's happy he's getting Keytruda. Here's the daughter. This guy looks like he's in a hostage video, but he shouldn't because he's getting some pretty good immunotherapy from Keytruda. So what I want to show you is it's not how silly some of these commercials are, but the applications for this one monoclonal anti-checkpoint antibody or immune checkpoint inhibitor. It is used now for all of these cancers, from skin cancers to cervical cancers to kidney cancers, and of course to triple negative breast cancers that have PD-L1. The point here being that the immune system is needed to fight, or is used to fight, virtually every kind of cancer. So anything that enhances the immune system is applicable to all of these kinds of cancers. And that in part is why at Find the Cause, we can study several different kinds of cancers in addition to triple negative and have results that are relevant to breast cancer. Here's a little anecdote about former President Jimmy Carter, demonstrating how powerful these antibodies can be. In 2015, in August of 2015, he was diagnosed with metastatic melanoma. So this is a skin cancer that has now metastasized to his brain. People with brain metastases like this aren't expected to live very long at all. At the end of August of 2015, he was treated with Keytruda, anti-PD-1 antibody. 4 months later, in December, he announced that he was cancer-free. So he couldn't find any cancer, including in the brain, by imaging. That's unbelievable. And this does happen to many patients, particularly with melanoma. It's not the average response. The average response is not that good, but this is the kind of thing that can happen when you unleash the immune system. You probably know he died last year in 2024 at age 100 from essentially old age, not from melanoma. And this is a guy who's relatively old. His immune system isn't great even at that point, and yet it still works when you take the brakes off. So what happens if you combine these monoclonals? Can you make things better? This is a chart of outcomes, so survival over months. If we're talking about breast cancer, melanoma, lymphoma, just keep in mind these studies are usually done with people that are pretty advanced along in their cancer. So let's look first at this melanoma, which is a skin cancer. So the average survival time with this aggressive type of melanoma is 18 months with just chemotherapy. And I'm gonna move something again. Okay, with just chemotherapy. If you give anti-PD-1, the average survival is 32 months. That's a pretty big improvement, almost twice as much. The anti-CTLA-4 compared to 18 months is not as great, but when you combine the two, anti-CTLA-4 and anti-PD-1, so antibodies that block two immune checkpoints, now the average survival is 60 months from 18. That's pretty good. For lymphoma in green, it's 14 months for chemo alone. At doubles with anti-PD-1. CTLA-4 just gives a little benefit, maybe, but you combine the two, and now 43%. The average survival is 43 months compared to 14 with chemo alone. Breast cancer in blue, follows the very same pattern, but it's not as dramatic. In part, that's because the immune system has got other issues with breast cancer that are maybe redeemable that we can fix. Now let me go to designer T cells, otherwise known as CAR T cells. Extra points for all of you who can tell who these fashion designers are. So what is this? So let me just tell you, just go back a little bit and talk about these killer T cells. So if a killer T cell has a receptor that could see some sort of mutated protein on a cancer. So Charlotte talked about this, that the cancers have got mutations, and usually different types of cancers can have anywhere between 50 and 200 mutations in their genome. Those will encode potentially for proteins that are different than any other protein that was expressed in that individual at any one time. So that would be, if these cells were okay, enough for the killer T cell to kill the target cancer cell. These guys are enormously efficient when they work. Let me just show you a video here of real killer T cells attacking a cancer. Moment.
These are cytotoxic.
Let's go back to the beginning. Go back to the beginning. Why don't you want to go back to the beginning? Let's try this one. Uh-oh. That's weird. Hang on. Try this again. There we go. Okay, so most of these pictures, green is the tumor cell. At one time they colored it red, but mostly, the T cell. This is the T cell, a killer T cell. And the red markings are essentially bags of toxins that the killer T cell delivers to cancer cells to kill them. So I'm thinking you'll be able to hear and see this.
Inside all of us lurks a serial killer. A killer whose primary function is to kill.
This is the killer T cell, this is the tumor.
Then kill again. These are cytotoxic T cells, a specialized member of our white blood cells. They patrol our bodies, identifying and destroying
This is a red killer T cell.
cancer cells. They do so with remarkable precision and efficiency.
So these are killer T cells floating through blood vessels in your body. They go everywhere, and that's how they get to find that there is cancer somewhere in your body.
There are about 5 million T cells in a teaspoon of our blood, engaged in the ferocious and unrelenting battle to keep us healthy. These amorphous blobs move around quite rapidly, pushing out their leading edge and probing their environment as they go. When a cytotoxic T cell finds
Again, T cell here, tumor here, and it's spreading its membrane out over there, sampling, basically licking the surface of the tumor cell.
membrane protrusions rapidly explore the surface of the cell, checking for the telltale signs of cancer. They kill their targets using poisonous proteins visible here in red. These cytotoxic granules move down special pathways in the cell called microtubules to the interface between the T cell and the cancer cell. The T cell punctures the surface of the cancer cell and delivers its deadly cytotoxins. This is very important in our bodies, where cells are packed together, as it focuses the lethal hit on the target and minimizes collateral damage to the neighboring healthy cells. The fate of the cancer cell is sealed. The T cell then moves on, hungry to find another victim.
And that last part is not just hyperbole. These are serial killers, which means after they kill one cell, they'll move on to another. Now, in the case of cancer, you have lots of killer T cells, and they have receptors, but the receptors are irrelevant to the cancer. They could be specific for some virus. You only have, again, a 10 or 12 maybe that could see the cancer at the beginning. So what you do is you redesign these killer T cells so they can see the tumor. So you bleed the patient, and then what you do is you take their T cells and you synthetically generate a gene that encodes a receptor for, let's say, HER2. So you remember that HER2 is a protein on the surface of the cancer cell. This gene will then encode a receptor for the HER2 protein. You put that gene into these killer T cells, it actually integrates into the DNA of these killer T cells. And since it encodes for a T cell receptor, it makes that T cell receptor that can see the HER2 on the surface of the tumor cell. Then you take that cell, grow it up like crazy in tissue culture, you end up with a bag of killer T cells, which are white, that you now infuse back into the patient, and now are, just can target the HER2 positive cells. Here's some data that show you a survival. This happens to be with acute lymphoblastic leukemia, or ALL. It's a very bad one to have. Most people don't survive 12 months. Maybe about 17 to 18% survive with chemotherapy. If you give the CAR T cells, you're up here in the 65% survival with a plateau that suggests these patients may be immune by virtue of these T cells for a longer period of time. These are old data really from generation 1 CAR T cells. There's not generation 8 or 10 by now. Each one has a different bell and whistle in it that makes the CAR T cell even more effective and able to live longer. And this little blurb down here just says many fewer side effects with the CAR T cells than the chemotherapy group. Now, what about cancer vaccination, cancer prevention? So we'll talk about vaccines, early detection, which means for us at Find the Cause, not detection of the cancer, detection of something that's changed well before the cancer happens to intervene before you ever get cancer. And we'll talk briefly about environmental chemicals. Now, if all cancers were caused by some infection, it would be relatively easy because a virus or parts of a virus, relatively easy to make vaccines against them. In the US, 4% of all cancers are directly caused by microbes. So it's not a lot, but it's some. Half of them from the human papillomavirus. And that means that if the human papillomavirus is involved, you should be able to make a vaccine against it. And you've probably heard about this. So this is the human papillomavirus. It is responsible for more than 97% of cervical cancers, anal cancers, vaginal cancers, and somewhere between 70 and 90% of oropharyngeal cancers. So oral cancers in the soft palate of your mouth. But you have now a vaccine, and have had it since 2006, called Gardasil. Now the Gardasil 9 has been modified to cover 5 different types of human papillomaviruses. If you immunize kids, basically before puberty or before sexual activity, you can prevent 97 to 100% of these HPV positive cancers. That's amazing. And the reason why you have to give it before they're sexually active, because the HPV is a cause of basically STD, and that result could be cancer. So the reason you have to give it before you are actually infected with HPV is because I can't think of a single vaccine to any virus or bacteria that's effective after you have symptoms. So once you have it, you can't, the immune response with a vaccine doesn't help you any. So it has to be given before kids are sexually active. Okay, now what about actual cancer vaccines against the cancer? Again, Charlotte and I just mentioned these mutations. You get these foreign proteins on the surface. These things should be recognized by the immune system. They're called neoantigens, and there may be as many as 10 to a couple hundred of these neoantigens. And then, in theory, you could make a vaccine to them by taking this, what looks now like a foreign protein, and injecting into the patients. Two issues here. First of all, you got to already have the cancer to know what these neoantigens are. So you really can't do prevention, but you could use these to prevent a relapse after patients have gone into remission. So that's one of the problems with this. It's not really primary prevention. The second thing is you probably still have to enhance immunity by giving some of these immune checkpoints. So I'm a believer that these kinds of vaccines have the potential to work in patients, especially when given in combination with the immune checkpoint inhibitors that take the brakes off. Okay, molecular biomarkers. So with apologies to Grateful Dead, trouble ahead. If we could identify somebody who is about to get cancer, we should be able to intervene with some of the immune enhancements that I've already talked about. So what does this mean? This is a cross section of normal breast tissue, where these are epithelial cells along a duct, an open area here, and these cells will now make milk that go into the duct that flow through to the nipple. If you get cancer, by the time you can image this, you already have somewhere near 1 billion cells in this area. So you're already behind the curve. Well before the actual cancer happens, there's something called cancerization of all the cells around in this area. And that means there are changes basically making the soil fertile that enable a single cell to transform into a full-blown cancer. Now, with this cancerization, which again happens well before the cancer appears, you have some telltale signs, such as the proteins that are being produced are different than normal. Genes are being expressed that weren't before. The metabolism of the cells around you have changed. So the Holy Grail, again, not the Monty Python top, but actually the really Holy Grail of finding these biomarkers is being able to find them in the blood, to be able to screen people with just a simple blood test for changes that might say you have cancerization going on here, you need to be treated with, let's say, an immune checkpoint or a different kind of a vaccine. And in finding the cause, we're spending a lot of time looking for these types of early biomarkers. So finally, I'm going to talk about very briefly primary prevention, which this is about. Okay, let's not even have this cancerization go on. Let's avoid it. And how do you avoid it? Well, you have to know how to avoid some of the environmental causes of cancers, and there are many. So pesticides have all been implicated in things like breast cancer, cancers like breast cancers. Herbicides like Roundup, don't use Roundup. If you do use Roundup, put some gloves on, please. PCBs, which are fairly ubiquitous in our environment, not as ubiquitous as PFAS, as the forever chemicals that I'm sure you've all heard about. Every one of us has got PFAS that's floating in our bodies, and they are probably carcinogenic. There are phthalates that are present in plastics that have been associated with breast cancer and other cancers. Air pollution, although it's gotten better thanks to the Clean Air Act, which is being compromised a little bit, and also heavy metals like arsenic and lead. And finally, solvents like benzene are involved in generating these cancers. So primary prevention and find the cause is don't be exposed to these things. And if you want to know how not to be exposed, how to avoid them, just go to the Find the Cause website and you learn about this primary prevention. So that's all I have to say. Again, thanks to the organization committee for the prevention party for giving us the time for this. And if there are any questions, fire away.
No question, David. I just say that the animation, not the animation, the actual T cell uh, Pac-Man
Yeah.
was very for me very illuminating, because I know you've spoken about these things in Charlotte and everybody else, and but to see it in action and might be an interesting premise for a short clip of it, of what we're talking about for the prevention party. But anyway, incredible.
Yeah, they are incredible. How do they do that?
I have a question that I don't know if you're going to be able to answer, but it's just come up in my brain as you were talking about this. Say we come up with a vaccine for breast cancer. How amazing that would be, right?
Mhm.
Do you think that the vaccine would be given before puberty, like a child vaccine?
I don't see that happening with what we know now, because as far as we know, breast cancer is not caused by some infectious agent. So if you knew it were caused by some infectious agent, you could take that virus or parts of it and immunize. That's basically what Gardasil is. But
Okay.
people have looked mightily for microbes associated with breast cancer and can't find it. Do they be able to immunize against something that's present in the breast cancer? You can't just take things that breast cancer cells express, because those are 99.9% self proteins, and there are no T cells or few T cells that could respond to that. So we wouldn't know what to make the vaccine against. We only know what to make it against when you have the cancer, and we can identify what are the mutated proteins, because it's only those mutations in the proteins that confer the foreignness that allow the immune response. So I don't see a pre-immunization with what we know now, or an immunization childhood or even early adulthood for breast cancer just now.
Thank you.
Yeah.
That was super informative altogether, both Charlotte and David. Thank you.
We do have a comment in the chat to repeat the slide to find out more info. I don't know if you can go back to that. So Google Pixel 7.
And I did reply with it at our website there.
Good.
It's findthecausebcf.org.
I have to just say that every time I see something that you're presenting, I pick up something else. And I don't know if I want to admit this, but I never realized that MAB in the end of something was a monoclonal antibody like aducanumab. So thank you.
I will also say the same. I always learn something new every time. It's so great.
That's
Thank you
great.
very
Well,
much.
I'll give you another little tidbit at the end. If it says MIB, it's an inhibitor. It's a small molecule inhibitor.
Ah,
There you go. That's the only thing
thank
you take
you.
away good enough. And but it blows my mind just seeing all of these things advertised. I mean, 20 years ago, inconceivable. And then it's just like every drug's a monoclonal.
So we have a question. Can you ask? Okay, Dan, I'm going to ask for you. What helps keep our immune system optimal? Is it water?
Yeah. Well, aside from avoiding the things I've talked about,
Yeah.
the things that are known to suppress the immune system are stress, through stress hormones that shut the immune system down. Poor nutrition, so balance your food. And there are some potential enhancers in natural products, but none of them have really been shown to make a really significant effect. As long as you have a pretty healthy diet, nutritional diet, it's not going to be boosted by some pill that you're taking.
But I just want to jump in and comment. Agree 100%, Dave, but I will put a shout out for vitamin D. Vitamin D is definitely
Yeah.
something that we're all deficient. A lot of us, especially in New England, are deficient in, and having optimum vitamin D levels does boost, immune health.
And exercise, right?
And exercise and
Yeah,
sleep.
Of course.
And sleep. Right. Well, it does. I mean, again, and there are physiologic mechanisms. It's not just magic. And getting sleep as well. All the things your mother told you turns out to be healthy. Never admit that though to her.
Here's an interesting question. Can we take blockers before cancer like Herceptin?
No, not like Herceptin in particular. For other, I mean, it will have some side effects. There are other cells in your body that have the HER2 on them. So you don't want to be messing with those if you don't have cancer. The immunoenhancers, like the immune checkpoint or the monoclonal antibodies, yes, but only with people that you know are at high risk. So for breast cancer, at least at this moment, that would be finding some biomarker that says that something is about to happen. At that point, you have to trade off a few side effects versus potential prevention. It could be very valuable, kind of hard to set up a prevention trial like that, but that could be very helpful. There's also other diseases where there are precancers that are actually, well, I wouldn't call them cancers, like monoclonal gammopathy of undetermined significance is a precursor to multiple myeloma. Somebody at high risk there might get an immunoenhancer. Actually, let me backtrack a little bit. Somebody who has some genetic mutation, the 5% or so that contribute to breast cancer, and they don't want to do surgery, well, maybe you want to enhance your immune system. It's not approved for that use just yet, but it's something to think about.
Can you become immune to the immunoenhancer?
That's a great question. So it's that would be possible if the monoclonal antibody was exactly the antibody that the mouse is making, because your body would see parts of that mouse antibody as foreign. But these monoclonals are engineered so that the mouse part of it is out of it, and the part that actually binds to whatever your target is still there. The rest of it is humanized. It's all human antibody on the other side. So far, no immune responses to the immunoenhancers that are monoclonal antibodies. That was one of the hurdles they had to get through.
So fascinating. Well, we are at 1 o'clock. Unless anyone has another pressing question, we're going to let these guys get back to work. Although Dr. Schur isn't really in need of that. He's retired. Still doing amazing work, especially for the consortium. Okay, looks like we're done with the chat, and I think we're going to sign off. Thank you, everyone, for joining us. Thank you, Dr. Schur and Dr. Cooper-Wasser. Amazing information that is just so valuable, and you made it really understandable, which is not an easy thing to do for lay people. We really appreciate that. So thank you, and we have a recording of this. We'll put it on our website. Please share. Please look on our website for additional webinars, which we have another one coming up on October 8th. And please look at our information about our event coming up on October 15th, our prevention party. It's going to be an amazing party that none of you will want to miss. Thank you, everyone.
Thank you.
Thank you.